Associate Professor Antony Cooper is with us today from Sydney. He's the head of the Neuroscience division at the Garvin Institute of Medical Research. He's also a cell and molecular biologist and geneticist with strong interests in shedding light on how cellular dysfunction results in human diseases. He has a special interest in neurodegenerative disease such as Parkinson's disease. His research on neurodegenerative disease focuses on understanding the basis of Parkinson's disease. Professor Cooper completed a PhD at McGill University and postdoctoral studies at the University of Oregon. As a tenured associate professor in Missouri, and since returning to Australia in 2006 to the Garvin Institute, Antony has focused his research on Parkinson's disease. So today we look forward, now that we're all energised and reduced our oxidative stress, he's going to talk to us about some of his current research, the current research directions that he's pursuing, and others more broadly. So welcome, Antony.
Thank you. I'm a scientist, not a clinician, and trying to understand what's gone wrong with Parkinson's disease so we can find out how to restore that or prevent it and all of those things. So I work at the Garvin Institute in Sydney. That's our famous staircase that looks like double-stranded DNA, and so we do a lot of DNA and genomics work, which you'll see coming up in my talk today. So there are a lot of, I'm not telling you anything new, there are a lot of big challenges in Parkinson's disease, and this is just a diagram that with time coming across here on the bottom horizontally is the progression of this disease, starting off with some less specific symptoms as diagnostic for the disease. Down here in the red line about perhaps when someone might be clinically diagnosed with Parkinson's, and then as the disease progresses with more time, those existing symptoms will probably get worse, new symptoms appear, and so this is, a major challenge. Breaking that down a little bit more is that if we're to find out, we need a better understanding of this disease, which is not telling you anything new because we don't know a lot about this disease. We've made some great discoveries, but we're still left with, in case if anyone's ever seen this type of diagram, 6 blind men touching an elephant, and what they're touching says, I think it's this. And so we have quite different, and yet none of them agree in terms of in this cartoon, which I'm simplifying. And what it's conveying is that we're making a lot of discoveries, but we don't quite have we've got jigsaw pieces, but we can't cheat. We don't have a box with a photograph and a cover for a jigsaw. And so we've got these individual pieces that all seem very important. We don't yet have that big picture which will be transformative for our really understanding of the disease. And regretfully, although as Steve mentioned this morning, there's some good indications of this drug exenatide having an effect, but we really do not have any therapies that stop the underlying disease progressing. Levodopa, etc., they are symptomatic treatments, brilliant in many cases. They are treating the symptom, but underlying that, unfortunately, that disease continues to cause degeneration and causing this progression. Another aspect is, as you can see in this diagram, we're diagnosing people and we're just at a guesstimate at this stage, maybe 10 or 15, as many as 20, it's going to vary on different people, later than perhaps when they first showed symptoms. So we're getting a late diagnosis. And obviously if we had a therapy, we'd be much better to give it to people much earlier on in the disease course. We just don't know about them. We can't detect them as yet. Secondly, as the earlier speaker this morning talked about misdiagnosis, when we're defining people based on symptoms, and they get, which is what is the case at the moment for Parkinsonism, then there are, just because there are other diseases with overlapping symptoms, there's some misdiagnosis. So, there's a fair amount of challenge there. Not to suggest we're of these colours, I just chose colours that would distinguish them with, I suppose, a Christmas flare with some blue thrown in. And that is to say, as again Steve Sant mentioned earlier today, Parkinson's is clinically a very heterogeneous disease. Some people have some symptoms, some people have others. It is mixed, and why is that? That adds further complication for us as myself as a scientist to understand what's causing this disease. If you have cystic fibrosis, it's got a mutation in a single gene. We have a fair understanding of that. We know what to target. When you've got a very common, complex disease that people get at different rates and different symptoms, that makes it much harder for us to try and understand, okay, what's going on here? And part of this may really be, and this is why I use the color, is that there are thought to be, at least on a clinical symptom base, different subtypes of Parkinson's. And that's been discussed for a while. As to why they might be, we do not know yet. So that handy question mark is to convey that there are probably subtypes. I'm not saying there are 3, this is just a hypothetical situation. And if there really are these different subtypes, we have to think about they may have different underlying causes. I would say, and many others would also, that Parkinson's might be this umbrella term of people with very similar symptoms. The analogy that I think is worthwhile to convey is something like breast cancer. It's defined as having a tumor in the breast, hence its name. But originally, drug treatments were never very successful for a large number of people. And until basically they were able to find that there are different subtypes of breast cancer, each with distinct problems that have gone wrong in that cell, genetically often, then you realize that, there are different subtypes. Each of those subtypes has a distinct underlying issue that's gone wrong and caused that disease. And in this case, if I was to use a color, that red might have one problem and it needs a therapy to address its individual problem. Green, subtype 2 and 3 might have distinct problems. They all might give rise to the same symptom group, i.e. breast cancer, but they have different underlying molecular mechanisms. And therefore, if we're looking for a treatment, we may find out we need to identify these different subtypes so we know there's no one fits everyone. We need to find out which group they're in, if that's the case. And I'm going to touch on that today. So given those challenges I just mentioned, my lab and others take different approaches. Mine I'm trying to think of as they're sort of integrated because they all come together as cogs, just as I've sort of drawn here. One was we don't really fully understand what's gone wrong in a patient with Parkinson's. And therefore, by trying to identify that will give us an idea of what is the disease mechanism. What are the molecular events that have gone wrong that have now caused a cascade of events to give us what we're calling Parkinson's disease? So we're looking to understand what's different, and that would be to give us insights of disease mechanism. If we understand the molecular problems, just like if someone just rings you up and says, my car won't work, but that's all you know, it's pretty hard to find out what the solution is. Someone's stolen the battery? Are you out of petrol? Who knows? So by understanding the disease mechanism, that leads us to the next thing. Well, if we understand the problem and identify it correctly, that allows us to now to think of what is the solution. So by identifying the disease mechanisms, that allows us to now to think of therapies that really get to that underlying mechanism. And what we're aiming for here is therapies that would slow, stop, or, if we're fortunate, reverse the degeneration and progression of this disease. And thirdly, though, as I mentioned earlier, we've got a relatively late diagnosis for this disease. If you're displaying symptoms a decade earlier, surely that would be the better time to give you a therapy, so if it slows or stops it, you never continue on and progress to the more severe symptoms. The problem is at the moment, those symptoms are not specific to Parkinson's. If I was to say to most people in this room, I'm constipated, I'm not sure if I smelled that pizza last night, that doesn't mean we all have Parkinson's, and that's our problem. Those symptoms are not specific enough. So we also need a way to be able to identify patients much earlier in the disease course. And this brings me to a thing called biomarkers. And biomarkers basically are indicators of disease, and I'll talk a bit about this today. And really part of my lab is to see, can we use something other than symptoms? Can we use biomarkers, measuring things in your blood, to detect patients who have Parkinson's much earlier in the disease course? It wouldn't just be useful, the other types of biomarkers, so that first one I was just talking about is can we identify, I'm not using the word diagnose, but let's just use that loosely here, can we classify someone and go, although you're not showing any distinctive symptoms, your blood work that we've analyzed in a very specific way suggests you've already started on that disease course. Secondly, we need another way of measuring where are you on that disease course. not just by a collection of symptoms, but by something that's more quantitative, more measurable. We can actually watch respond and see if it goes up or down, and therefore that helps us that if we put you on drug X or Y, not for the symptomatic treatments, but to slow progression, are they working? And that's all part of also monitoring a disease patient in terms of are you progressing or not just simply relying on the clinical measures that we're using so far, but have a fair amount of variation in them which causes problems. So let's say we got all these things, how's that really going to help? If we could diagnose people early, we've understood the mechanism, and now we have a therapy. Well, hopefully I don't really need to explain that to you, but here's that starting slide. So for existing patients, if we found therapies that would stop progression or slow it, then we would arrest those people at the symptoms they have and prevent them from progressing or getting worse. For people who have just recently diagnosed, then again, we could give them therapy, these type of things to stop progression, and that would stop them at those earlier symptoms, and they too wouldn't progress and have further and further disabilities. And then what we're really looking for the patients of the future, and unfortunately they are coming, as we all know, it's a conveyor belt at the moment unless we find therapies. That is, could we identify patients early and give them that therapy and stop their progression then? Then they would never really advance to those motor symptoms and those more serious non-motor and consequences of that. That's kind of what drives me and others in this Parkinson's field to look at that. But not surprisingly, you're probably going, so how do you find someone before they have symptoms? You crazy scientists with black magic. That's not the case. Well, we might be crazy scientists, but we're looking for approaches that are going to get us earlier diagnosis. And how will we do that? Or how am I doing that, and how are others? I've just come back from a clinical meeting in Hong Kong of international specialists in Parkinson's, and that's a huge theme of we need biomarkers to be able to find people earlier, to measure their progression, because if we measure their progression, we can find out if drugs work. At the moment, we're in a bit of a bind there. So how do we find biomarkers that are going to, before you show symptoms? Well, the type of research, and this tells you how research can then have clinical applications, is we were looking for the disease mechanisms in Parkinson's patients, as in what has gone wrong or what is not working properly in the brains of people with Parkinson's, because if we can understand the difference between them and people of a similar age who don't have Parkinson's, surely that should give us an indication of what some of the things that have gone wrong, and therefore give us an idea of therapy. So just to be clear here, in Parkinson's, maybe 5 to 10%, depending how we're cutting that cake, have problems in a single gene. It might be one of 20 genes, but they have a genetic underlying mechanism that out of the 20 so Parkinson's genes, as we call them, a patient might have a mutation in one of those, and the sum of them all might account for 5 to 10% of the patients. But for 90 to 95% of patients, they do not have a mutation in the gene, in those 20 Parkinson's genes. And they're referred to, in that case, as idiopathic or sporadic, which is a polite medical term to say, we don't know. And that's their subtlety there. So there's no clear disease mechanism, and they're clearly a contribution from genetics. It's just not that they have one gene with a problem, but it's complex. And that's what's held up our advances. Now there's also environmental impact. And so if you look at this, as you heard earlier, there's both genetics and environmental impact, and the relative interactions of those two are going to be very complex. But because most of the patients are idiopathic, that's what I chose my lab to go after, to find out what is dysregulated in the brains of idiopathic Parkinson's patients. How do you find out what's gone wrong in someone's brain? Now, first of all, if I was asked for someone for a sample of their brain right now, they would perhaps be reluctant. It's called patient reluctance for some reason. We've yet to crack why that is, but it's quite clear. And therefore, we have some challenges. Secondly, it's one thing to say, oh, have a look, find out what's different, but I mean, that's like looking up at the stars. And what do you measure, or what do you look at? So to give you an idea here, this is genetics. I'm hoping this is showing up here as the DNA. Now, DNA is sort of, as you've heard, the blueprint or master copy of all our instructions. And those instructions, if you want to think of them as blueprints in an architect's office, they're cocooned nicely and held safely in the nucleus, and then you make temporary copies of that, and that copy or temporary copy is called RNA. And that RNA then is a molecule, kind of like a photocopy of the blueprints taken out to the construction yard, and is used to assemble the 20,000 or so different proteins that are active in all of your cells and allowing us to be what we are. When you want to know, we have about 20,000 genes. When you want to know what's different, one of the ways is to say, okay, let's look at a group of Parkinson's patients and a group of healthy individuals of a similar age who don't have Parkinson's, and let's, as one way to try and understand what's going wrong, go, what genes are on in the Parkinson's patients? What genes are different, turned on or off? Are they up more or down less compared to those healthy controls? So the way to do that is the amount of RNA that is present from any one gene is an indication of whether that gene is on or off. We can do a very sophisticated approach where I'm at the Garvin, where we can basically count each of those RNA molecules from samples of people's brains that have been donated kindly post-mortem of Parkinson's patients and controls, and work out, okay, gene 1, that's the same in patients and controls. Same, not of interest to us. Gene 2, this level, patients, this level. I'm interested in gene 2. Go through all 20,000 of those genes and work out what genes are what's called dysregulated. Up on too much or not enough, and give us some insights then as to what are those genes, what are the proteins they encode, do they give us some insights as to what has gone wrong in, what is dysregulated in the brains of idiopathic Parkinson's patients. So we did that, and I'm not going to get into the heavy science here because I'm sure there'll be some reluctance, and that's fine, you can ask me later if you want. But what I wanted to give across is we've identified changes in the RNA of different genes in the brains of patients. What we did then was also go, how many of those genes can we detect in the blood of Parkinson's patients? And are they also differently up or down compared to healthy controls? Because obviously we can't have someone coming in at 40 and we take a brain sample and go, let's just check if you have Parkinson's disease based on RNA stuff. What I'm looking for are biomarkers that are far more accessible. Everyone's used to giving a blood sample, that's not too tough. So I wanted to see whether could we find that the changes, some of the RNA changes that are happening in the patient brains, are they also seen in their blood? Because that would give us biomarkers we can screen far more readily and avoid the inappropriate brain request. So that's what we did. We identified a large number of RNA changes in the patient's brain. That has given us some insights to the disease mechanism. That's pretty specific and hardcore science that I didn't think would suit today's audience. But in addition, it also allows us to look for biomarkers. Can those RNA changes in the blood help us say, this person has Parkinson's, or we think it is? Remember, our aim here is, can we identify people who we think have Parkinson's before they get clinically diagnosed by a GP or neurologist? Could we use this to screen people at 40 and say, or 50, and go, you have indications of Parkinson's based on a blood test? I'm not going to go into all the machine learning and algorithms we did, but we have some pretty nice preliminary data that in a blinded samples where we're given a number of samples and not told whether they're from healthy individuals or people with Parkinson's, could we distinguish them into two groups? And we had pretty good success with that first group. And the US NIH, National Institute of Health, are just sending us about 300 more blinded samples to test us whether will these biomarkers really, can we really distinguish people with Parkinson's from those without? Now, this would be basically us here taking people we know already have Parkinson's. So more about 5 years post-diagnosis. If we can then show that we can detect those, we get access to far more valuable samples, which are harder to get, blood samples from people who have just been diagnosed. They're very valuable and hard to get those samples, so they don't want to risk them on just anyone's whim. They need to have proof. If we could then say, yes, we can detect people who have been recently clinically diagnosed, then we can access some very rare samples of people who had given samples a number of years before they developed Parkinson's. Could we have spot them there? If we can that, then we're going. Now we've got a sample from a blood test that to see whether can we detect you even earlier? And that if we have a therapy, those two go together like a hand and glove. Just to give you an example of what that use would be, and this is a completely hypothetical situation. This is looking at a number of brain cells or neurons, and it's completely me just drawing a cartoon on the plane this morning. And this is where perhaps an average person who's not going to get Parkinson's with aging would have a slow decline of those neurons. And for some reason, and I didn't have a nice curve, but people who are going to have Parkinson's, maybe they at a certain age start to lose the function of those neurons. And finally, about this stage, drop below a threshold level where they have insufficient function, and now they'll start to show symptoms. They may present themselves to a doctor, and then let's just say they get clinically diagnosed, let's say 63 here. But if we could identify those people 15 years earlier, instead of diagnosing them at, say, 63, our biomarkers allowed us to detect them at 50. We currently have federal bowel cancer screening that I think kicks in at 50. Maybe this could be something if they work. If we detected that someone had markers in their blood that they have started on their disease course, and we have a therapy that slows it, then maybe we could, this green line, just slow the decline with therapy, having detected them earlier, and this is a hypothetical situation, delay the onset of those symptoms, so that instead of occurring at 63, in my completely hypothetical situation, it slowed progression that they're symptom-free until 80, which is a long time to avoid those symptoms. Now, in addition to the biomarkers in the blood through RNA profiling to identify, do you have Parkinson's, and also to stop the misdiagnosis is another important thing, early and accurate. There's also, we need to measure progression, not just on your sort of symptoms, but a quantitative measure. That's going to have a lot of uses. So as I said, biomarkers are useful for monitoring of disease. And some of you may be quite familiar with giving a blood test to see whether do you have diabetes. So here's another indicator of a blood biomarker to help monitoring a disease. So this is an HbA1c monitor, and basically it shows you how well have you controlled your blood glucose levels, nothing to do with Parkinson's, well, not seemingly. How well have you controlled your blood glucose level over the last 3 months, which is the lifetime of those red blood cells. And if you end up, depending on the scale you use, that you're a 5 or 6 or 5, pretty good. 6 or 7, okay, there's a number you're getting quantitatively, bit of a worrying, maybe more exercise, less stuff. And then if you're starting to get into high numbers like the red, right, let's give you a therapy. Let's see if that helps maintain your blood glucose. So it's also telling you your disease status, but just as importantly, if your doctor puts you on some drug to help you with metformin or something like that for type 2 diabetes, you can come back 3 months later and see, hey, I was in the red at 10. Now with this drug, I'm back at a 7. That also tells us the drug is working for you. And that may sound simple, but that's also what we're looking at for Parkinson's as a biomarker for progression. And here's just again a very simple, very simplistic example of where this is in this case with progression and time going on for a Parkinson's patient, the upward arrow is showing that it's getting worse, but we would be able to get more accurate measurements. Where are you on the disease course? Think of that diabetes test. Can we have a numerical system to go, where are you? Are you progressing or not, versus less definitive techniques? So that's helping your doctor monitor you, but also given what I'm going to talk about in a few minutes, it's really important in clinical trials for drugs that are going to slow progression, because we need some sort of quantitative measure more than the clinical measures that are used by your neurologist or GP, so that we can actually see, does a drug work? So if there's a clinical trial to test drug X, at the end of it, we need some measure to be able to go, people who are on the drug are doing better than those, or have not progressed, or have less progression than those not on the drug. And that may sound like, well, surely we can tell that, but the amount of variation in our clinical measures we have when you see the neurologist makes it hard to go statistically, which is the basis of a trial, to go, yeah, statistically, this drug really has helped. If those clinical trials are not quantitative or measured enough, you could end up with a strange situation of perhaps having a drug that probably does work, but you can't tell. So these biomarkers, again, have a really strong need. So that's the biomarker section, and I'm going to bring all these things together at the end, but I'm just switching then to the genetic contribution to idiopathic Parkinson's, which I said patients do not have a single Parkinson's gene mutations. And yet clearly all the other measures, and I'm a geneticist as well, is suggesting genetics is playing a role. So how does that reconcile? And so by sequencing the genome of patients, which is where we take the DNA of people and do sequence the DNA, all 3 billion bases, if you're doing a whole genome sequencing, start to see that, and this is preliminary data, so don't ask me further about this, this is just us cautiously talking here, is that some patients seem to have a combination of genes in certain pathways that we would think would be to do with Parkinson's, but instead of having one devastating single mutation that destroys it, they might have several mutations that affect all the other things around it, like the satellite cities. Rather than a strike on Sydney, there are a number of mutations on the satellite cities that surround it that would also cause perhaps a similar problem. So this is not familial Parkinson's where there's a single gene inherited. This is raising a very attractive possibility of that a number of idiopathic Parkinson's patients, the genetic component might be that they've inherited 4 small mutations. Any one alone might increase their risk, but together, if they've been unfortunate enough to inherit all 4 from their 2 parents or have a de novo brand new mutation, presents a situation where they're at much greater risk due to their genetic contribution. So if we then are able to sequence patients, obviously with consent because there's a huge privacy issue, then finding out those genes of the idiopathic category will give us a whole range of really crucial information. It will tell us that that individual has this problem over here, just like that breast cancer aspect. They perhaps need a drug that works for this. It would give us a huge understanding of Parkinson's. It would give us, like a really revealing, transformative perspective as a strong possibility to go, this is what this disease is about. It's a cluster of people getting different mutations in different parts of their cell, and that is why they get Parkinson's. And what's even more exciting for me as a scientist in the hope to bring therapies to patients is here was this clinically heterogeneous disease. Everyone acknowledges that. Are there subtypes? People certainly see subtypes in terms of symptoms. But our collaborators in the US have, and this is not for distribution, I'm just bringing it to you, so please don't go out and tweet it and everything, since you all look like heavy tweeters, is that by starting to sequence some very preliminary data, and that's why I don't want any hype in it, and it's not my work, it's our collaborators, is that there are indications that this heterogeneous group may break down into those groups based on or somewhat that may be a genetic basis. That subtyping might be, if you're sequencing patients, you may start to see them. Some of them have a problem in this part of the cell with this pathway, others over here. Think of the analogy to the breast cancer as one that works quite well. And this just offers us some tremendous breakthroughs if that's really the case. We might find out that really there is proof that there are subtypes, that Parkinson's is an umbrella term for a syndrome or a collection of different sort of more discrete diseases. We could find out that if there is a drug, it might help the people in subtype 1, but not 2, because they have, in essence, underlying differences. I'm talking big blue sky picture here, please. We might then find that, look for targets. For if subtype 2, we know that where their problem is in that pathway, and we could look for repurposed drugs that have been found for other diseases that hit that same target and go, let's use that on Parkinson's patients of subtype 2. So this is wrapping up in terms of we need therapies to stop progression of the disease urgently. And that involves clinical trials of drugs. So I've been involved with a large group, all coming together, both nationally and internationally, to set up, to do clinical trials of some drug. We don't have funding yet, so this is all awaiting, so please don't, I'm trying not to get ahead of ourselves. Here we are looking to test a number of drugs or repurposed drugs for slowing progression of Parkinson's disease. But as I've conveyed to you, just doing clinical trials by themselves, we're going to run into problems. And this is why we're going to have these problems, and this is why I've spent the start of the talk talking about biomarkers and genomics, because they're going to come together with the clinical trials and hopefully give us the best case scenario. I talked about misdiagnosis. We don't want people who don't have Parkinson's being tested for a drug for Parkinson's. It won't work for them, and that will confound or confuse our clinical study. We need blood RNA biomarkers to go, do you really have Parkinson's, or do you just have symptoms that overlap? We need to just have people with Parkinson's in our clinical trials. We need to be able to measure more accurately, has the disease progressed? If you are on this drug, have you progressed more slowly than people who are not on that drug? And we need quantitative measures to show that statistically. We're confounded that what if, like breast cancer, Parkinson's really is several different diseases with the same symptoms. We won't, like with the breast cancer, one drug will not help them all, so it'll look like a failure. So simply just having a clinical trial without these components is going to really reduce our chances of success. And inversely, having solutions to these problems will really enhance our chances of success. And that's why these blood RNA markers for diagnosis, progression, and our genomics integrated seamlessly into the clinical trials is really what we're looking to have. So this is a large integration. I've been working on this for over 2 years, as for others. I'm more the scientific lead, not the clinical. But what it is to bring together clinical trials in Australia of predominantly repurposed drugs and integrating it with biomarkers and genomics so we can understand and interpret this as best as possible. Just finishing, here would be in a case where in a clinical trial you might have a drug and let's say 15 people out of 100 respond well. In a clinical trial, what do you do with that information? Do you give everyone that drug? Pretty expensive to give everyone that drug, and it may have side effects without any benefit. So we're left with this sort of, what to do with that? But one of the reasons for integrating genomics into this is if we find that there are genetically defined subtypes of the disease, now you might find that, when I put on that decoder ring and look at this, those people who responded, they're all of subtype blue, or whatever my numbering system was. Now we suddenly switch to, wow, we've got a drug that might help people of that subtype. And we know how to screen the rest of the patients in Australia to be you of that subtype. And we could go, this is big blue sky, if you're of subtype 1, we think we might have a drug for you, at least to go into phase 3 trials. If you're of subtype 2 or 3, or green and red, do not take this drug. It will not help you. It might have side effects without benefit. That's also appealing to the pharmaceutical benefits system that they are only giving money towards drugs that are beneficial, because obviously Treasury always has a big role here. So I just wanted to touch on that, where we're bringing these 3 different components together. And if you were to look at this in some case, if you look at the bottom, if we can basically diagnose people earlier, genomically define their subtype, and have a matching therapy for that subtype, we have the real chance of treating people today and in essence preventing them suffering this disease in the future, what I sort of call a functional cure. So thank you for your time, and I appreciate your time.